Welcome to Lecture 7 on the topic of an introduction to soils and agronomy. This is part of the Agronomy 1 subject, which is offered in the agricultural degree at North Melbourne Institute of TAFE, an education institution based in Australia. For any details about our educational products, please visit our website at www.nmit.edu.au. My name is Dr. Nikki Cooley. During this lecture, I will introduce to you soil and some of the components and concepts around soil. We'll start with an introduction on soil. What is soil? How is it formed? We will then look at why it is important to agronomists and plants. I will introduce some key components and characteristics of soil. These include texture, structure, pH, cation exchange capacity and fertility. Please note you will study other subjects as part of this degree, which will examine these concepts in more detail. The objective of this lecture is to give you an overview. So let us start by introducing some simple concepts. Soil is an essential resource for the production and growth of plant and crops. It is a mixture of mineral, organic matter, organisms, pore space, liquids and gases. It is formed over long periods. Its formation is part of an action of chemistry, of physical interactions and of biological factors on sedimentary igneous and metamorphic rocks. For an overview about how soil is formed, please follow the internet link on Moodle to watch the following video. This is an APES video on soil formation. As demonstrated in the APES video on soil formation, there are 5 factors that affect what a soil is like. The first is the parent rock material, and this largely affects soil chemistry. The second is the interaction of temperature and water. The third is topography. Usually, the steeper a hill or mountain, the quicker the soil or rock will erode. Fourth is the diversity of organisms that you have in your environment. And the 5th factor is time. The soil profile is a vertical section through which the soil extends from the surface to the unweathered rock beneath. Soil can be divided into layers, and these layers are called horizons. On this slide we have 2 images. On the left-hand side you see a hypothetical horizon map, starting from the surface litter going down to the bedrock. On the right-hand side you can see a picture of these different horizons and what they look like. The O horizon is the surface litter. This is where you have fresh or decaying organic matter, for example leaves, twigs, crops, and these accumulate on the ground. Horizon A is known as the topsoil. This is the top layer of the soil, and it holds most of the organic matter and most of the water and nutrients that is required for plant growth. Infiltration and seeping of water down through the soil starts at this layer. Horizon E is the zone where leaching occurs. Horizon B is the subsoil. This is the zone that often contains an accumulation of minerals as well as some organic materials. Horizon C is a zone composed of broken down plant material and the basic type of rock of which the rest of the soil tends to be derived from. This is unless the soil has moved. You will note that roots typically are found on the sublayers sometimes, but mostly going from the topsoil through to the subsoil, that is layers A, E and B. Soil scientists have developed soil classifications that parallel the plant taxonomy. A summary of these soil classifications and where they are located in Australia is presented on the slide. This image was from the journal article by Mackenzie et al. In 2004. You will see that there is quite a range of different soil classifications across Australia. One such soil type from the Australian soil classification is called podosoil. The image on the slide from the Australian Soil Club illustrates what a podosoil looks like. Podosoils can be found in Victoria. For a full description of all Australian soils, please refer to www.soil.org.au. The link will be located on Moodle too. Soil contains organic matter. The amount of organic matter that a soil contains can vary largely. Organic matter is important as it is a source of nutrients for plants. It also has other functions. It aids soil aggregation, it also aids soil structure, and it provides storage for water. So why is soil so important for crop growth and agronomists? Well, soil has many functions for agronomists and crop growth. Principally, it acts as a medium for the production of crops. This medium allows roots to grow in it. This serves two functions. The soil can act as an anchor for the root and also an environment for the root. This environment aids the absorption of water, oxygen, and essential nutrients. The ability of a soil to aid water, oxygen, and essential nutrients is referred to as the soil fertility. All of these components are required for plant growth and development. Soils also enable the medium for the symbiotic relationships with such organisms such as nitrogen-fixing bacteria. It also aids as a storage of water between rainfall events or irrigation. When managing your land, it is easy to become focused on the role of soils and crops with respect to their environment and their ability to anchor roots. But it is essential that you remember that soils are fundamentally ecosystems, and quite complex ecosystems at that. They contain numerous organisms that interact, and these organisms include plants, bacteria, fungi, insects, and invertebrates. Within the soil's ecosystem, there is a continued cycle between prey and predator. In order for you to fully understand some of the concepts of soil and ecosystems, I'd like you to watch the following YouTube video on soils and ecosystems. This was produced by USDA. When you are watching this video, please ensure that you make notes about the different types of soil organic matter that are described, and about hydrological functions and why they are important in these forests. Essential elements are found in the soil. They are necessary for a plant to complete its growth cycle, whose function cannot be replaced by other elements, and that are compounds of a molecular or enzyme within the plant. Minerals that are derived from rocks in the soil provide many of these elements. In most plant species, there are 17 elements that are essential for plant growth. The compounds such as oxygen, carbon dioxide, and water are all obtained from the atmosphere. Plants acquire the other essential elements from the soil. There are several classifications of plant nutrients, but the most commonly used is that based on the content of the nutrient found in the soil. These are separated in either macro or micronutrients. Macronutrients, i.e. compounds that are found in large scales, include nitrogen, phosphorus and potassium. And it is these compounds we often add as fertilizers to the soil to aid our growth and development. You will learn more about this topic in future lectures. Another important concept to understand is that of the carbon and nitrogen ratio and how it relates to decomposition in your soils. The carbon to nitrogen ratio is the relative amount of carbon to nitrogen. It influences the rate of decomposition of animal and plant material in the soil. The C/N ratio varies among plant ecosystems. Therefore, if you add different conditioners such as soil humus or compost, or you leave young alfalfa on your fields, you will have a different carbon to nitrogen ratio, which will significantly influence decomposition. Plants with a low C/N ratio, such as alfalfa, are nitrogen rich and they decay rapidly, while plants that are high in the carbon to nitrogen decay slowly. An example of this is sawdust that has a 400:1 C/N ratio. The reason for the importance of these ratios is that the bacteria and the fungi that decay these plants require nitrogen for growth. If inadequate nitrogen is available, the number of microorganisms will be insufficient to decay the plant material. When carbon to nitrogen ratios are more than 30:1, all of the available nitrogen in the soil can be used to sustain existing microbe populations. Therefore, if you add sawdust to your soil, it can result in your crop becoming nitrogen deficient, and additional nitrogen may be required. When considering adding organic material to your soils, whether it be animal or plant waste, manure, hay, etc., be sure to understand and find out what the carbon to nitrogen ratio is and how that will impact on decay, and therefore how it may impact on your crop nutrient requirements and your crop nutrient management. Particles of decomposed organic matter are very small, yet they have a relatively large surface area that is chemically reactive. Overall, organic matter has a negative charge. This large surface area in the soil helps attract the water and nutrients, and thus influences what we call the organic matter fractions. That is, all the organisms that make up organic matter. They include such organisms as earthworms, potworms, anthropoids, nematodes, mites, colymebia, protozoa, ciliates, fungi, and bacteria. As you will see from the slide table, their approximate length ranges quite significantly, as well as their biomass. Individuals can range from 2 to 3 to billions per 1,000 square meters cubed. The fractions of organic matter have many roles. These roles include sequestration of nutrients, that is, such as carbon. As carbon is a major component of the organic matter is therefore an important component of the global carbon cycle. Organic matter is a source of nutrients. It acts as both a reserve for the essential nitrogen, phosphorus and boron, but also nutrients are attracted to the surface of organic matter. Nutrients may be a component of the organic matter too. Organic matter is a binding agent that joins the mineral proportions of the soil together. The aggregation of soil particles is important for the soil tilth and aeration. These are important for successful plant growth and development. Organic matter absorbs water like a sponge and it retains it for plant growth. And finally, organic matter can tie up toxic chemicals and pesticides that would otherwise contaminate the groundwater. This is referred to as chemical sequestration. In the organic matter fraction live the humble earthworms. A good population of earthworms indicates a healthy soil. Earthworms are small animals that can improve the soil structure by contributing to physical breakup of the soil. Some soils can contain up to 300 earthworms per meter cubed. There are 7,000 species of earthworm. Across these species there are 3 classifications: the litter dwellers, the shallow soil dwellers, and the deep burrowers. They play several critical roles in good agronomical soils. They shed crop residues and initiate decomposition of the organic matter by bacterial and fungi that feed on the organic matter. Their extrae is in turn consumed by the soil organisms. They mix soil and organic matter, and they bury soil organic matter. They allow tunnelling to occur within the soil profile. This adds aeration and provides pathways of movement for water and plant roots. Let us now look at the soil properties that affect plant growth and crop growth: texture, structure, and chemistry of the soil. That is, such things as pH and cation exchange. Soil texture defines the soil fineness or coarseness. A soil can be very fine or very coarse, or somewhere in between. It is determined by the type and the amount of specific soil particles. These are soil, silt, and clay. Sand and silt are principally silica, while clay, being the smallest compound, is comprised of a mix of compounds which include silica, aluminium, oxygen, hydrogen, potassium, magnesium, and phosphorus. It is a chemically reactive particle and provides most nutrients for plant growth. Agricultural soils tend to be a mix of sand, silt, and clay. Principally, the soil is composed from sand, silt, and clay. Once you know the composition or the percent of each of these components, sand, silt, and clay, you can then use this information by using a soil triangle, the figure on the slide, and this you can then identify what soil type you have. For example, a soil with a high clay content, low sand and low silt is described as clay. However, a soil with an equal amount of clay, silt and sand is called a clay loam. Soil structure describes the clustering of soil particles into various shapes. Productive agricultural soils often have a granular structure in which clusters of soil particles are bound together by organic matter and clay. Well aggregated granular soils have good aeration and tilth. Tilth refers to the soil having beneficial qualities related to crop growth. Soil structure can be defined by the volume and the arrangement of these pores in the soil. This determines concepts such as soil-water movement and air flow in the profile. This affects soil friability, that is, the ease of rain infiltration and rain runoff, cultivation and growing crops. Damaging soil structure can be an issue that you'd like to avoid. Livestock can pulverize the soil, that is, they can result in the destruction of large and small soil channels. These channels allow the rapid air and water infiltration. Wet soils often result in compaction and breakdown of soil aggregates if livestock are run on them during this period. Binding organic matter is very good at binding and breaking up the soil structure. Soil structure erosion. Low organic matter causes collapses in wet soil conditions, reduction and infiltration, and an increase in runoff. 5 to 10 times more erosion when poor soil structure exists. There are a number of clues that can lead you to identification of poor soil structure. Look in areas that you suspect you may have a problem and compare them with a similar soil that you suspect has good structure. For example, before livestock were grazing. Rill and sheet erosion is where water runs over the soil surface, strips away thin sheets of soil, only millimeters at a time. When the water collects, small eroded channels form down the slope. This is rill erosion. Surface crusts reduce infiltration to water and restrict seedling emergence when they become dry. Cloddiness. After a single cultivation, brick-like clods are formed. Further cultivation is required to produce a suitable seedbed. Some plants can be used as an indicator of bad pore structure. One such plant is the toad rush. It grows where water has ponded on the surface. Ponding is where water after a light rainfall accumulates. It does not soak into the soil. A hard surface may be seen with poor soil structure. This is when the soil is dry and tests the hardness. The depth of penetration of failing mattock or with a pick can be used to test this. Compare different soils. Poor infiltration may result after the first. This is when, after the first rains, after a dry spell, water does not infiltrate into the soil very far. And finally, the presence of organic matter. Soils tend to be brighter and paler in colour when they do not have much organic matter. When there is a large concentration of organic matter, the soils tend to be dark. So what course of action can you take if you suspect or find poor soil structure? You can change your management to a permanent pasture. Plants will protect the soil from erosion, and the deep roots from the pasture will help the pores and channels needed to improve infiltration, and plants will naturally increase the soil organic matter. Manage the pasture well, however. Ensure that you use water, nutrients, and light grazing wherever possible. The application of the compound gypsum can be used to improve your structure. If you apply gypsum, it is best to tackle slacking and dispersion of your soils. The rate of gypsum that tends to be recommended is 2.5 tonnes per hectare. Gypsum works by supplying calcium, which disposes the sodium from the clay surface. It is advisable to test for your exchange cation levels, and this can indicate your application rate. We will also look at soil chemistry. Soils have many chemical properties that are important for plant crop growth and development. The three that we're going to concentrate in this lecture are cation exchange capacity, pH, and salinity. Small crystalline clay particles and organic matter that exist in the soil are chemically active and usually have a negative charge. The negatively charged surface of these particles enables them to attract positively charged particles. If you are able to link to the following website, you will see a demonstration of the chain exchanges of these charges. These images are on a conceptual nature. The amount of exchangeable cation a soil can sustain is called the cation exchange capacity. Where this is relevant for you as an agronomist is that the higher this cation exchange capacity is, the more able the soil is to hold nutrients and is therefore fertile. The cation exchange capacity for the soil particles clay is 30 milliequivalents per 100 grams, for sand it is 9 milliequivalents per 100 gram, and humus has a high level of 200 milliequivalents per 100 grams. From these three components, this shows you that adding humus to the soil will increase your cation exchange capacity. There is a range of exchangeable cations found in the soil, and some are listed on the slide. Their ability to attract varies also. That is, aluminium is very attracted or strongly associated with the soil, whereas sodium is less so. Knowledge of your cation exchange capacity in your soils will enable you to manage your soil environment correctly. This is particularly important if you are going to add supplements to the soil, and it is also important in your nutrient management of your soils. Acidity is the chemical condition of the soil and is measured by the pH scale. The pH scale starts at 1. This low number represents a strongly acidic environment, and it ends at 12. The high number of 12 represents a strongly alkaline environment. Acidic soils have a pH of 4.5 or below. This is when the pH is measured in the soil solute calcium chloride. Soil pH can also be measured in water, and you need to be mindful of how it is measured in the interpretation of that pH. Acidity and understanding your acidity is important because it can reduce yields and also interfere with nutrient availability and nutrient uptake. Australian soils have a tendency to be slightly acidic or acidic. There are several consequences of acidic soils. It can prevent legumes from nodulating. It results in a reduction of recycling of your nutrients by beneficial soil microorganisms. It can increase toxicity to your plants by two methods. One is the release of aluminium from the soil, and two is the release of high magnesium levels. High magnesium levels are typically seen in hot, dry, poorly drained acidic soils. If you have low pH, you can have a decline in nutrient availability. Available aluminium and iron may tie up valuable phosphorus, for example, or it can cause molybdenum deficiency. There are a range of effects of acidic soils. Different crop and pastures are affected differently by different aspects of soil salinity. Soil salinity can affect the top 10 centimeters, and it tends to be where you see the problem. However, the problem may occur at the subsoil level as well. Subsoil acidity is costly and very difficult to adjust for. The diagram on your slide describes the influence of pH on soil fertility and nutrient availability. If you start by looking at this figure at the pH 7, which is regarded as optimal, you will see after studying this slide that all of the nutrients can be taken up. If, however, your pH is above 7, it is regarded as alkaline. alkaline toxicity can occur. The more alkaline the soil, the more toxicity results. If you have alkaline soils, you can get deficiencies of iron, manganese, zinc, copper and boron. You will tend to see an excess of sodium, which can be toxic to the plants. If you reduce your pH and you are entering acidic soils, as your pH reduces, your toxicity increases. If you have acidic soils, you can have deficiencies in magnesium, calcium, potassium, phosphorus and molybdenum. You may also see an excess of aluminium, manganese and iron. This demonstrates the importance of agronomists to understand your soils. Add your nutrients to your crop. You could at worst be wasting your time if you get your nutrient-soil interactions incorrectly. You could also result in a significant loss of yield, nutrient deficiency, and toxicity issues to your plants. So what are the signs of soils that are acidic? You tend to get uneven pasture or crop growth. Pay attention to acid-sensitive plants, and you may see the invasion of weeds that are acid tolerant. Acidity across a field tends to vary significantly. This may match your uneven pasture variation. You may see poor nodulation of legumes. Stunted root growth and increase in root disease may also occur. If you find that you are having difficulty in certain crops, such as lucerne and medics, this may suggest soil acidity problems. And finally, you may see the formation of organic mats on the soil surface as a result of the loss of biological activity. So what is the action that you should undertake? Well, if you suspect soil pH issues, the first thing to do is to conduct soil pH tests and aluminium level tests. Remember that soil acidity varies greatly due to rain and temperature over the season. So when you are conducting your soil samples, not only should you take a close note of the climate and conditions when you take the samples, but also that you can compare these conditions at similar times. Ensure that you understand the difference between measuring pH in water versus measuring pH in calcium chloride. It is often thought that the measurement of pH in calcium chloride is more accurate and more meaningful for agronomists. 20 or more samples should be collected from a paddock due to the large variations seen in acidity. There are a number of actions that you can take if you suspect acidic soils. The most common is the addition of lime. A heavy application may be as high as 6 tons per hectare if you have a seriously acidic problem. The lime needs to be spread evenly over the affected areas and ideally incorporated into the soil. This is where managing subsoil acidity can become very tricky. The lime works by changing the pH and neutralizing the surface. As a rule of thumb, the guide of lime to add is 2 tons per hectare for every 1 pH change you wish to undertake. Ensure that you measure your pH after your lime application. The lime application may depend on the soil type, as clay loam soils tend to need more application than sand soils. It is best that you add your lime at 1 ton per hectare every 8 weeks following the initial application. Continue this for the rest of the season. You can also look at the method developed by G. Fenton in 1993 about strip application of lime. This is where a strip of lime is applied on the field and you monitor the performance of the plants over time to see if this lime has made meaningful differences in your plant growth and development. Another action that you can consider is to do nothing. This is not advisable, however, because plant yields will continue to decline even if you do not see this initially. Another action could be considered is to grow more tolerant species. If you do this alone without trying to adjust for the pH, this is probably only a short-term fix, as your pH or your soils will become more acidic. And finally, the fourth action that you can consider is to change your farming system. If you reduce the intensity of your cropping, for example, and you lightly graze your paddocks, you may increase your acidity. There's been experiments where lightly grazed undeveloped pasture in acidic soils only needs rates of lime application as little as 0.5 tonnes per hectare every 10 years. Intensive production systems of high crop yields, such as hay cutting areas and productive pastures, need about 3 tonnes per hectare every 10 years. You should also consider better nitrogen management, better soil moisture management, and an increase where possible of perennial pastures and deep-rooted plants if you have acidic problems. Soil salinity is also called saline sodic and saline sodic, and these terms are used interchangeably. Salinity occurs due to the accumulation of salts such as sodium, calcium and magnesium that dissolve in water. As the water table moves, so does the salts. These salts form a chemical reaction between acid and a base. Most soil salts are sulfurs, SO4, and the chlorides, Cl2. Salinity tends to be associated in hot, dry environments due to the higher evaporation of water from the soil's surface. There are two types of salinity, dryland and irrigation. Salinity reduces yields because salts cause plant dehydration. The image on the slide was taken from the Mallee region in Australia by the Department of Primary Industries. It illustrates the effects of salinity. It is toxic to plants. Salinity may cause waterlogging issues. When water is in the soil, it is not used. Deep drainage to the water table increases. Native trees have been found to be very effective at using this wastewater, and removing them reduces the capacity and leads to salinity issues. Annual crops and pastures tend to be shallow-rooted in nature, and they do not use deep water, this also leading to salinity. Overgrazing can reduce leaf area and lowers plant water use. Sowing late can also cause or make salinity problems worse, as if the paddock is not using the water in the autumn, this can again add to the problem. Disease and inadequate fertilizer, or any factor that limits plant growth, can also impact on salinity. Where you allow long fallow, you aim to store water, and often the soil is full before the crop is sown. Additional rain recharges the groundwater, adding to your salinity problems. Leaking dams are an obvious one, but little thought about. And finally, poor surface drainage. Water laying on the land powers plant growth and water use. This then leaks into the water table if it's not used up. So what can you do if you suspect salinity problems? Well, the first thing to note, it tends to be a whole farm or catchment approach. A farm plan is highly recommended, and the Department of Primary Industries can assist you with such a plan. Ideally, you aim to use water more efficiently. You can do this by planting native trees and shrubs, changing your cropping practices to perennial pastures that are well managed. High water use cropping, early sowing varieties, and avoid disease of these varieties. You need to consider good crop rotations and allow a longer pasture phase by using perennials and avoid long fallows. Surface drainage should be repaired, and use groundwater irrigation for stock, for example. I'd like to bring this lecture to a close by looking at erosion and conservation of your soils. Erosion is the accelerated loss of soil through the action of wind or water. Management techniques such as conservation tillage and growing permanent vegetation to cover your soil can minimize this erosion. A useful tool in conserving soils and preventing erosion is being able to estimate ground cover and then manage accordingly. Note that ground cover does not include bare or loose soil or clay pans or scales. However, ground cover does include live plants, sticks and logs, litter and dead plant material, rocks and stones, dung, soil crusts able to withstand a hoof impact, and plants such as lichens. This slide shows two images obtained from DPI. One on the left-hand side demonstrates 20% ground cover estimation, while the one on the right-hand side shows 80%. To avoid soil erosion, you should aim for a 70% or more cover on your soils. And that brings us to the conclusion of the lecture on an introduction to soils and agronomy. I hope that you now have a better understanding of soils and their importance, that you are able to define soils. I hope that you now have an appreciation for managing soils means managing an ecosystem. This ecosystem is complex. Soils are indeed essential for good crop growth and development. Many farmers have stated in the past that if you are able to successfully manage your soils correctly, obtain a good soil fertility, the rest is easy. Ensure you understand the concepts of structure, texture, and chemical properties of the soil and how you need to use this information to manage your crops appropriately. Ensure that you are able to estimate ground cover and understand this relationship with erosion.